This video presents declassified and licensed material of the Global Institute of Logic and Cybernetics.
Livestock accounts for approximately 14.5% of global greenhouse gas emissions. Because these supply chains are bound to massive tracts of arable land, they remain highly vulnerable to climate disruptions and geopolitical choke points. Mitigating the inherent unpredictability of climate-dependent agriculture requires a transition towards deterministic engineering. ScrollFusion, Rodopa Ethical, is a distributed, gravity-independent biomanufacturing asset designed to execute this transition. It operates as a sovereign node that produces high-volume protein without requiring land usage, insulating the food supply from environmental volatility. ScrollFusion functions as a cyber-physical biomanufacturing system, utilizing established process analytical technology loops and built to overconform to European Food Safety Authority standards. Replacing biological stochasticity with engineered replicable precision provides the mathematical baseline for protecting national food autonomy. This schematic outlines the deployment vessel, the ScrollTower. It is a 12-floor urban manufacturing facility housing 192 modular reactors. At steady state, a single pilot tower is engineered to produce 800 tons of biological output per year. To bypass energy costs of pumping viscous nutrient media upwards, the ScrollTower utilizes gravity-assisted media return lines. These flow downward from the sterile reactor floors through the central core. Eliminating high-pressure return pumps caps energy consumption at 0.55 kilowatt-hours per kilogram. Substituting mechanical pumps with architectural gravity transforms an energy-intensive process into industrial infrastructure. The ScrollPod is the atomic manufacturing unit inside the tower, constructed from 316L stainless steel. Standard bioreactors face scaling limits because they use gas bubbles to oxygenate the fluid. At industrial volumes, physical shear stress from these bubbles destroys cell viability. The ScrollPod bypasses this failure mode using silicone and PTFE membrane oxygenators lining the inner walls, paired with a low-shear, magnetically coupled impeller. This configuration achieves a mass transfer rate of 15 per hour, governed by deterministic AI control loops adjusting feeds dynamically. Eliminating the physical variance inside the reactor ensures the predictable, linear scaling required by institutional capital. ScrollFusion relies on an approach where flavor and texture are treated as structural memories, stabilized epigenetically over time. Optimizing for immediate yield via genetic edits inherently triggers premature contraction programs, which results in a disorganized cellular architecture. This split screen compares the results of genetic speed optimization against constraint-based epigenetic engineering. Standard fast growth optimization creates an isotropic structure, a disorganized mass that lacks mechanical resistance. ScrollFusion instead subjects the cells to continuous mechanical tension within a precise calcium to magnesium ionic ratio. High magnesium levels stabilize the chromatin energetics. This forces the cells to mature slowly, resulting in the formation of true anisotropic fibers. We engineer flavor by navigating a mathematical manifold defined by mineral exposure, cumulative time, and oxidation. Replicating the mechanical resistance and breakdown sequence of traditional meat provides the sensory checksum human memory uses to verify food authenticity. This physical alignment allows the product to fit within existing culinary frameworks without relying on artificial additives. Because the ScrollPod utilizes bubbleless mass transfer and magnetically coupled mixing, its core bioprocessing mechanics are gravity-independent. In microgravity, the complete lack of convection causes gas bubbles to foul sensors and creates free-floating cellular aggregates. The ScrollPod Space V1.0 pressure vessel is an orbital hardware module designed to solve these aerospace bioprocessing challenges. To prevent cells from floating freely, the module uses a nanofiber matrix, relying on capillary anchoring to physically bind the cells and nutrients in place. The control system uses triple modular redundancy microcontrollers, protected by aluminum lithium and polyethylene shielding, to withstand orbital radiation. This module maintains a 92% cell viability rate, even in simulated microgravity conditions. Extreme environment viability validates the terrestrial architecture as a dual-use asset for national space programs. To stabilize facility economics while awaiting human-grade regulatory approvals, initial production volume is routed through the ScrollPet operational wedge. Producing feed-grade protein utilizes the same biological process, but operates under existing animal feed regulations, generating immediate cash flow to cover fixed operational expenses. The kernel stack governance layer protects the intellectual property and revenue models of this network. A physical firmware lock ensures ScrollPod bioreactors cannot initiate perfusion without a digital authorization token. Real-time telemetry is routed through a zero-knowledge proof system, matching physical production to authorized revenue tokens. Cryptographically linking biological output to digital licenses makes IP theft physically impossible, securing the sovereignty of the asset. Translating the blueprints into the Sofia facility requires a €50 million CAPEX. At scale, this targets a €25 per kilogram OPEX, with 45 to 55% margins. By year 5, a tower projects €22 million EBITDA, a 3.2-year payback, and a 30% IRR. Following the pilot phase, this 12-floor blueprint will be replicated via licensing across energy-advantaged regions globally, operating as a replicable infrastructure asset class, avoiding the risks of continuous biological redesign. This model positions national food security as a deterministic equation based on industrial capacity and engineering precision.